So today you guys are going to be doing a candy chromatography lab like I told you yesterday. And there's lots of different supplies you're going to need, as you can see over here. You're going to eventually need 4 different colors of, I think Mr. Hackett got you M&Ms. You're going to need a pencil. So I do not want you to have a pen in the lab at all today, because otherwise somebody's going to forget. So you need to have a pencil, no pens in the lab. Okay, it can be a mechanical pencil or a wooden pencil, it doesn't matter, but it has to be a pencil. You're going to get a glass petri dish, so those, make sure you get a glass petri dish that does not have a bunch of wax on it from Candle Labs. Remember, just look where the frogs are, it'll tell you where they're at. Distilled water we're going to use today, which are in the little squirt bottles that are found on the counter. Hopefully Chell got the salt solution made that I emailed him to take care of this morning. Which should be in a giant beaker, and then you're going to need a 250 milliliter beaker. I don't know why I said 125, but let's use the 250 milliliter beaker. Those seem to work the best. And you're going to need something called 4 capillary tubes. Capillary tubes are little teeny tiny pieces of glass tubing. And we'll talk about those later. And then you're going to need a ruler at some point. You don't have to take that into the lab with you, but you can. And when I say ruler, I don't mean a meter stick. You want something smaller than that. Okay, so what you're going to be doing is you're going to be testing how far the different candy dyes will move up the paper when you put the chromatography paper into solution. Now remember, what this does is it's going to separate out colors, and the colors are going to move different distances based on if they're attracted to that saltwater solution more or if they're attracted to that chromatography paper more. So what we're going to do is we're going to go through these steps, and I have a little place over here where I can draw to show you what we're going to do, because I would normally be doing that on the whiteboard for you anyway. So the first step is you're going to select 4 different colors of candy to be tested. When you do select candy, if there are M&Ms in there, I want everybody to get at least one, or not at least one brown. You need to get one of your colors to be brown. Okay, the other 3 M&Ms should all be a separate color. So one should be brown, and then you can pick red, orange, yellow, green, blue. I don't care which ones you get, just pick one. Okay, but they should all be different. So you should have 4 different colors. One of them should be brown. You're going to place the candies in a petri dish as far apart as possible. So shut my slate off. So if this is your petri dish, okay, you're going to put, let's say, like a red candy here, okay, an orange one over here, your blue one here, and your green one over here, okay? And by the way, one of these should be brown, remember, okay? So one of those should be brown, but you're going to spread them out on a petri dish, put them in the 4 corners of a circle, if that makes sense. And then you're going to make just a few drops of water so they have little pools around them. Make sure those pools that are around them don't touch. So don't put huge squirts of water, just a couple of drops, a couple of drops, couple drops, couple drops. The water shouldn't go out farther than, right next to it. Okay, so that's not going to take very long to do, but while somebody is doing that, the other partner is going to go get a piece of chromatography paper, and you're going to have to cut it to a specific size. So if you have a beaker, okay, here's my beaker. Oop, let's make a better beaker. That's just quite awful. Erase. Okay. So if this is my, wow, you know what? We're just going to have an awful beaker. Okay, there's my beaker. If I have a beaker that looks like this, my paper is going to be just smaller, like just skinnier than it. Okay, so that when you set it in your beaker, it can actually like stand up. Okay, and you can easily grab a hold of it, and it doesn't like slump at the bottom too much. Okay, you don't want a lot of slump, so it should look like this from the side, not a whole lot of this. Okay, so this is what we want, not that. Hey, you're going to cut out a piece of chromatography paper like so. By the time you get that done, whoever set up the candies, you can go ahead and take the candies off, because your water should have taken off some of the dye that was on the candy. Now you should have little colored pools. Okay? So when you get your chromatography paper, let's go ahead and, well, here, let me make this bigger, drink, so that I can undo. Let's just undo these. Okay, So you're going to get your chromatography paper. So like this, okay? You're going to, actually what I want you to do is you're going to do number 5, or sorry, do number, where is it? Do number 4 before you do number 3. So go ahead and do this one first. So do this one, okay? So you're going to cut a piece of chromatography paper. Then you're going to do number 4. So on your papers right now, I want you to like draw an arrow or something for number 4 up above number 3. That way you remember. So fill some beaker up. You're not going to fill it up, okay? You're not going to want very much. So if this is my beaker that I drew instead of my chromatography paper, you're only going to have, you know, maybe this much filled with salt water. Okay, not much of all, not much at all. You're going to put your paper in it, and you're going to see what I mean here in a little bit. So, if that is your beaker, okay, you're going to take your chromatography paper. Since I ran out of colors, I'm going to use purple as my chromatography paper. Okay, so you're going to just set it off to the side. Okay, so you're going to do this to the side. You're going to like stand it up over here, and you're going to make a mark with your finger that's like, oh, say about here. Okay, that is just slightly above the line of salt water that you put into your beaker. Okay, so wherever you put your finger, it needs to be above, just above where your salt water solution is. Then you're going to put, like, just lay this piece of paper on the table, your chromatography paper. Wherever your fingers, you're going to draw a line in pencil. Okay, draw a line in pencil straight across. Okay, it's very important that that line is above the solution. Okay, that's why I was talking to you about the slumpiness of your paper and everything else. So this line needs to be in pencil, and it needs to be just above your saltwater solution you have poured into your beaker. Okay, once you have done that, you're going to take your capillary tubes. Okay. And you're going to take them and suck some of the water up. Now this isn't going to be like, oh, I'm going to take my tube and I'm going to sit it down in the water and wait a very long time. Okay, you're going to get too much liquid really doing that. What you'll do is these little capillary tubes use cohesion and adhesion properties of water to suck it up really quickly. So you're going to take it, you're going to have it straight up and down, and you're going to just set it in one of the pools of water. When you see that you have some in the tube, you're just going to pull it up. It'll take like a half a second, okay? Just a quick little dip down, pick it straight up, okay? Then you're going to come over here to your chromatography paper right away, and on the same side that you used, you're going to put that capillary tube right on that line, okay? And let's see, we were doing red, okay? We're going to put that red dye right there. You don't need a very big dot, okay? That's a plenty big dot. Remember, you're going to need to have 4 of these going across. So you're going to do that, and then once you're done with that capillary tube, once you're done with that color, you're done with that capillary tube. So then you're going to pick up the next one, get some color into it, put it here. Okay, put it down. Get the next capillary tube, put some color in it, put it there, and so on, so that you have those. Make sure that when you're doing that, they're not going to absorb into each other like as it spreads out. You don't want them to absorb into each other. It could potentially ruin what you're doing, okay? When you're done with your capillary tubes, you can just go ahead and throw them into the glass bin. I mean, it was just water, so there's no chemicals in it, but we still don't want to have them get broken in the trash. So throw the capillary tubes in the trash can right away when you're done. All right, so you have this. Now you're going to simply take this paper, okay, and put it into your beaker that has your solution. So make sure, whatever you do, make sure that your paper, or this line on your paper does not get into the solution, okay? Keep it above where the solution currently is. Okay, so when you set it in there, or you might want to hold it in there just to be safe, you're going to notice that the saltwater solution is going to start climbing the paper right away. Okay, and you're going to leave it in there for several minutes. It might be 10, 15 minutes or so. Sometimes it's 5. It's random. I don't know why it's random, but it is. And what you're going to see is when the solution has climbed so that your chromatography paper is wet to, like, let's say here, okay, just a little bit, like a few inch or so from the top, it's probably going to be done. You're going to notice that it doesn't move anymore. Once it gets to a certain spot on your paper, you can be done. Okay, so that's what you're going to do. You're going to then see how these little blotches that you have are going to move up the paper with a salt solution. Different colors will move different amounts, or at least they should. Sometimes it works well, sometimes it doesn't. Remember over here, the smaller the dot, the better. Don't let them blend, because if they blend, it's going to get all weird. So the smaller the dot size you can make on here, the better. Okay, so when you take it out of the solution, you're going to make observations and record the quantitative data in the table provided. So what you're going to do is once it's all done and it's not sucking up any more solution, then you're going to take this paper out, and let's say this red one's moved to here, okay, this green one moved to here, the blue one's like here, and the orange one's still just like there. Okay, what you're going to do is you're going to find the center of the dot, of the splotch, wherever that is, of each color. You're going to take that center and you're going to measure from there to the line. Okay, this is called like an RF factor, and we're just going to call it the distance traveled though. You're going to measure that in centimeters and write it down. So the dye color, I'd say blue, and then maybe this distance is like, I don't know, a half a centimeter. You'd put 0.5 centimeters down for that. Okay, and you do that for each color. Then you're going to use that data later to help answer your questions. One of your partners, I realize both of you can't do this, so one of your partners is going to take your chromatography paper and actually tape it to your lab paper. Make sure it's not soaking wet when you do this so that your entire paper doesn't get wet, but it should dry pretty quickly. Tape it there. So I can see it, and so that I can see that you actually measured correctly. Once you have that data recorded from each one, so remember center of the dot all the way to the line, you're going to go ahead and answer these 4 questions about it, and that'll be all. So you are going to have those 4 questions for your lab due tomorrow. The rest of your lab hopefully shouldn't take too long. I'll be interested to see what your papers look like, since I'm not here to help you through it, because a lot of times 2 or 3 groups have to restart every time, so we'll see how it works out. But good luck, have some fun. Remember, you no pens allowed in the lab today, only pencils, and you may not eat any candy that was in the lab. Got it? Don't eat any of it. Don't dump it out onto a table. Just don't eat it. Don't touch it. Only take what you need. Okay? All right.